Hi there and welcome to this A-level biology video with Mr. Phillips, and today I'm recording from in my classroom, so apologies if the sound is slightly lower than normal, but I wanted to try and get these done for you. So today we're going to be looking at the circulatory system, and effectively we're going to look at what a double circulatory system is, what adaptations each type of blood vessel has and why. We're going to look at pressure changes as blood flows through the circulatory system. We're going to look at calculating the area of a blood vessel and how you would do that, a bit of maths. We're looking at tissue fluid and calculating the movement of tissue fluid and how it's formed. And we're going to look at, really briefly, at what the lymphatic system is. So just to introduce it, the idea is that we all need, as multicellular organisms, to have mass transport systems. And this is because multicellular organisms have a low surface area to volume ratio. Now what this means is that we cannot supply all our body cells with diffusion alone. So if you've got your cells, whether it's your arms, there we go, okay, and we want to get the nutrients into, let's say, the middle of your arm, that distance would be too great in terms of diffusion difference. So we need a constant supply of oxygen for aerobic respiration, nutrients, again, for different parts, different functions, and different, maybe aerobic respiration again. Amino acids, obviously make proteins, fatty acids, and also we need a rapid removal of carbon dioxide. And the transport system in your blood obviously helps do this. And obviously the diffusion would be too great, it would be across too larger distances to meet the metabolic demands of cells. So it ends, in this case, at the end of the route, at the end of your transport system. So your oxygen, for example, diffuses in via your lungs, goes through your blood to your respiring tissue system. So the human transport system is a double circulatory system, and it's important to understand that this means that the blood travels through the heart twice for every passage, every circuit of the body. So if we start at the, let's say the kidneys, okay, the blood travels from the kidneys and back by the vena cava to the heart, and that blood has got very little oxygen, it's deoxygenated. It then travels from the right side of the heart into the lungs to pick up oxygen, and then from the lungs via the pulmonary vein back into the left side of the heart, and then through the aorta back to the other parts of the body. And that's really important because it's travelling through the heart 2 times at every circuit. And there are some blood vessels that you need to be very familiar with from this diagram. So you need to be familiar with the pulmonary artery. So the pulmonary artery is the blood vessel that goes from the heart to the lungs, okay, and it carries deoxygenated blood, which I'll just write deoxy there, okay. You then need to know about the pulmonary vein, which carries the new oxygen-rich blood from the lungs to the heart, and that is going to have lots of oxygen in it. So that's the only vein that carries oxygenated blood, just like the pulmonary artery is the exception to the rule for the arteries, and it's the only one that carries deoxygenated blood. The main artery leaving the heart is the aorta. So this diagram here shows the aorta splitting up to go up and down the body, and it carries the blood from the heart to all parts of the body, and obviously it gets shorter and splits into different vessels as you go from it. The main vein coming back to the heart is the vena cava, and it's split, as you'll see in the later lesson, into the superior vena cava, so into 2 parts. And also what you should have done in the kidney lessons, or you will do in A2 in the kidney lessons, is there is also the renal artery and renal vein, and they supply the kidneys with oxygenated blood and also takes the carbon dioxide away from the kidneys, and that's one that you also are expected to know. So blood vessels then have this sort of overriding structure. These are, some of these are A-level pictures, some of these are GCSE pictures, because you've done all this before. So you can see here you've got an artery, we've got a narrow lumen in the artery which helps maintain pressure. We've got elastic fibres which will expand, but they'll also recoil, and that helps maintain the pressure of the cell. And then you've got a layer of tough collagen fibres and muscle fibres on the outside. The vein, however, even though it's carrying the same volume of blood to the heart as has left the heart, the vein has a large lumen, and therefore the vein will be under low pressure as a result, because it's more of a diameter. And the vein also has valves which prevent, if the blood's going that way, they prevent the backflow of blood. And then the capillary here, which is a lot smaller in terms of its actual size, being magnified there. The capillary has got 1 cell thick, and the lumen is just big enough to squeeze a red blood cell in. So that means when red blood cells flow through there, there is very little diffusion distance to get across that 1 cell thick into the respiring tissues or into and out of the alveoli, for example. And that's the adaptations that you really need to know. So you need to know that the arteries carry normally oxygenated blood, and it's at high pressure away from the heart. The endothelium, which we often don't talk about, the inner lining is folded. That allows it to stretch, to get bigger during the heartbeat as it pushes more heart blood out. Elastic tissue recoils, so that means it pulls inwards in order to, as the heart beats, and that maintains the pressure. And as I said, apart from the pulmonary artery, it is always oxygenated blood. Now what happens is the veins, the arteries, get to the organs that they're on the exchange site, and they split into smaller arterioles so they can completely surround the organs and areas of exchange. So the arterioles are when the arteries split into a network of smaller vessels, and one of the key adaptations of the arterioles is that they can, the muscles in the arterial wall can contract, and that can constrict the arteriole, preventing blood flow through certain arterioles. Now if we just go from this, imagine you've got one vein and that splits into several arterioles. One arteriole then will split into maybe thousands of capillaries. So if we have a look at capillaries, first of all, as we said, capillaries are 1 cell thick, and they've just got a lumen just wide enough to allow red blood cells to squeeze through them, which decreases the diffusion distance. And it's the endothelium of the capillary that is 1 cell thick. And we say there is a network of capillary beds where there is a lot of capillaries. Then the capillaries all eventually rejoin to make what would be called a venule to start off with, but then a vein. And a vein, as we just said, has got a large lumen, which means that it can have lower pressure, and it has valves to stop backflow. And apart from the pulmonary vein, all veins carry deoxygenated blood, and all veins go towards the heart. So I always tell people to remember A for arteries, away from the heart, and the veins go into the heart as a result of that. Okay. So here we have a typical graph which shows you the pressure changes that happen in the blood vessels as they leave the heart. So there's a couple of really important points to identify here, and that is if we sort of label them here as A, B, C, D and E, and obviously that is showing us the arteries splitting into the arterioles, into the capillaries, and into eventually the venules to go to the veins. So the blood vessel here would look something like this. You'd have a big artery, okay, that is splitting into the arterioles, as I've said there, okay, so imagine they're splitting into that. That splits then into lots and lots and lots and lots of capillaries, like that, okay, and then they rejoin, and as I said, we don't really need to talk much about venules for our A level, but we'll just say that like that, okay. So what is happening here? Well, first thing is, you can see there is an upwards-downwards pressure. So the upwards pressure is when the heart is contracting and it's forcing the blood out of the heart. And as you will be aware, that's when the ventricles contract to force the blood out of the heart. That causes this increase in pressure in the arteries as more blood flows through. And the arteries are adapted to allow that. And they're adapted to allow that because the elastic tissue in the arteries can expand, and then it can recoil when the blood vessels drop, the blood pressure drops down. And the other thing is the endothelial lining, as we've said a few minutes ago, is highly folded, so that's fine. Now as you get further away from the heart, the blood pressure starts to drop very, very slightly because of the distance, and there is a resistance because the blood is flowing and it is coming into causing friction with the walls of the endothelium. So whilst they are smooth, there will be some friction starting to happen with the walls. Now when you get to the arterioles, there's an increased cross-section and an increased friction with all the different walls of the arterioles, and again the same happens, so you get all this extra friction with all the extra walls and the narrowness of the capillaries, which lowers the pressure right down because the capillaries are very, very narrow, and we know that the veins have very low pressure to go back, and there are valves in the veins to ensure there is no backflow. And the blood is brought back to the heart in the veins due to breathing, and that lowers the pressure in the veins when you breathe out, and also it's due to muscle contractions which force the blood forward. And now this could be a question that comes up in a maths question, and it could link to the surface area of the red blood cell, or it could link to the surface area of the cross section of a blood vessel. So what we have to do is remember this, which is the GCSE maths equation, area equals pi r squared. So we're going to assume that pi is 3.14 for this question, okay? And let's have a look at the question. So the question says that a blood cell has a diameter of 7 micrometers. So you need to know that diameter is the full distance across the circle. So the half the distance, which is what we're using in this equation, is radius. So that will be 3.5 micrometers. So therefore, I'm going to do pi r squared, okay, which is pi, which is 3.142, and I'm going to multiply that by what we're doing, so pi r squared, so the radius is 3.5 squared, okay? So let's have a look at that. So what I'm going to do is 3.5 squared, so 3.5 times 3.5, which equals 12.25, multiply that by pi, and that equals 38.49 micrometers, okay? But, and this is quite an important but, surface area. So we're assuming that the cell is flat, but if it's got flat, it's got two sides. So I then have to multiply that by 2 to give me that answer there. I don't know how likely that's to be as a question in an exam, because it is a bit strange saying it's flat. But what you could easily get is a question like this. So this one is a bit more possible, and it's calculating the cross-section areas of blood vessels. So again, you've got to do pi r squared. So in terms of doing pi r squared for this one, we look down each one. The artery, we've got to work out the radius of each of them. So we're going to be doing 52.6 divided by 2. So that equals 26.3. 19 divided by 2, which equals 9.5. 3.7 divided by 2, which equals 1.85. 21 divided by 2, which equals 10.5. And 76.2 divided by 2, which equals 38.1. So then what I need to do is do that radius squared and then multiply it by pi. So if I just do one of those to show you how I would do it. Okay, so again, all I would do is pi r squared. So pi, we said, was 3.142, and I'm going to multiply that by 26.3 squared. 26.3 times itself. Okay, so if I do that, I'm just double checking for you on the calculator, that equals 21,070. Yeah, good. Okay, so those are the different ways. So again, you've just got to do that. Remember, you're using that, but the radius is half the diameter, and that is really critical that you don't forget that to work that sum out. Now, what that leaves us with then is the only other part we need to talk about is, well, what happens at the exchange sites? So let's say you've got a capillary bed that has come round into the muscles as has been shown here. Well, the cells, it's not like individual cells only line blood vessels. So what happens is there's lots of gaps between cells, okay, and those gaps then are basically in a bed of fluid that can flow around them, allowing exchange to happen. So as the blood comes in from the artery into the arteriole and then splits further into the capillary, due to the high pressure still, or the higher pressure, smaller molecules are forced out into this bed of fluid and to form tissue fluid. And they can freely diffuse, and it's small molecules such as amino acids, oxygen, carbon dioxide and water. Larger molecules, particularly proteins and red blood cells or white blood cells, cannot diffuse out due to their size. So some fluids leak out into the tissue fluid, and that becomes tissue, into the space between tissue, and that becomes tissue fluid. Now we have to control this, so this point here doesn't mean that water cannot go through, but it has to be controlled, because obviously if all the water went out, this would cause you a big problem. So there is a sort of comparison sum that you need to understand to do this. So here we are at the arterial end and the venular end. So we've always talked about this arteries, about having high pressure, and we've always talked about veins about having low pressure. So whilst we're in the arterial and venular end, we are near the high or low pressure. So as we come to this bit, and we could use some numbers to show this, so let's use some very simple numbers. Let's say the pressure, the hydrostatic pressure is, and I'm making these numbers up just to show, 5 kilopascals. Okay, there's a high pressure because you're near the arteries. There will be some reverse pressure trying to push the liquids back in, okay, and that is osmotic pressure. So there will be some osmotic pressure, but this is very small, so let's say it's 1.1 kilopascals. We'll explain why that's small in a second. Well, that 1.1 kilopascals means that overall this side has an outwards movement of, can't do my maths, 3.9 kilopascals. Now as we get further along, there will be a lower pressure. So let's say the pressure drops to 3 kilopascals, okay, and the osmotic pressure, let's say it could be very much the same, or it could be slightly higher now. So let's say the osmotic pressure is, I don't know, 1.8, okay. That then means that there's less of a difference between the two, and in fact the osmotic pressure might actually become greater. Let's say it's 3.8, and that would move then 0.8 back in. So it's how the tissue fluid goes back out. Now, why is there a difference between the osmotic potential of the artery end to the vein end? Well, if you think about it, the water hasn't left here, so the water in the blood, there's high water levels, okay, and that water then is going to move out, so you've got a higher water potential as a result here. But once we've got all that water out and there's more water in the tissue and there's less water and there's more proteins in the venular side, this side will have a lower water potential. So a way to look at this could be something like this. Okay, so here's a sum that we have here, and we've got showing you what is happening. So we've got the hydrostatic pressure, and the difference between 4.3 and 1.1, okay, is 3.2. The osmotic pressure, okay, it goes from minus 3.3 to minus 1.1. The osmotic pressure is that. So you just do 3.2, take away 2 equals 1.2. But on the next side, on the venal side, we have some different numbers. We've still got in this example the same osmotic potential, minus 2, okay? But in this case, we've got a lot less pressure. So 1.6 here, 1.1 here. So we've only got a pressure of 0.5. So if we've still got an osmotic potential of minus 2, so an inwards movement of minus 2 and an outwards movement of 0.5, overall we're going to be going in by minus 1.5. 0.5. Now, what all that does, it makes our makes lymph, okay, or the tissue fluid. And what we're saying is not all of that tissue fluid can get back into the vein, into the capillaries to get back into the venules. So therefore, some water has to enter into the vanule via osmosis, but excess fluids, ones that when we can't get it all back in, enters something called the lymphatic system. And what the lymphatic system is effectively a series of ducts, and they have other roles including controlling fat and excess levels of things like that. But basically they run alongside, and in safe areas where it's safe to do so, run alongside then your blood vessels, and that's where the excess capillary tissue or lymph tissue at this point goes back